Welcome back. It's lecture 5 in the membrane series. One of the first key experiments looking at membrane proteins was actually done by researchers studying red blood cells, trying to understand their structure and function a little bit more. And what they found was cells could be lysed under hypotonic. So go back to your high school here. Hypo means water, fluid moving in. Hyper, you're sweating, you're losing it. Iso means no net movement in or out. So this is the red blood cell. You harvest these, put these into a hypotonic environment, and what that does, it fills these cells up and they burst and they lyse, and usually lyse at one position. And if you wash, you end up getting leaky ghost cells. In other words, kind of assuming the original structure, but there's kind of a hole in it. Okay, and so now we have access to the outer end. The blue is the inner leaflets. You can wash and then change the environment, and you can get your ghost sealed. You can disrupt sonication, whatnot, disrupt and reseal, and you'll end up getting a mixture here of vesicles, small vesicles from the red blood cell that are right side vesicles and inside vesicles. So we have the outer leaflet here open to the surface and the inner leaflet in these cells. So we have 4 different types of remnants of cells that we can do studies on. Okay, you're probably seeing where this is going. So when they did a total protein profile and one-dimensional SDS, this is in stain, looks like this is a radioactive labeling of proteins and then exposing it to X-ray film. They found a series of proteins that were always found, and under Kamasi blue, they found the spectrum molecules. We talked about that in our cytoskeleton, alpha and beta, a protein known as ankyrin, band 3, glycophorin, band 4.1, and actin. And here's the molecular weight in Daltons. Okay, Daltons, DA. Okay. So an amino acid equals 330 Daltons. Okay, you'll need that for biochem, that conversion. Here's our ankyrin, our spectrum, alpha and beta. The beta has a series of phosphorylations that allows it to dimerize and interact. And they found that the spectrin, alpha and beta, are 25% of the protein's mass in one red blood cell. So 1/4 of the proteins in red blood cell are spectrin. So right away, when something is that abundant, it's probably there for a reason, and that maintains the structure and the biconcavity of the red blood cells. So it takes a lot of this protein to actually make those red blood cell structure. And the spectrin twist, little 2 Twizzlers, interact with other proteins. There's some actin, adductin proteins, and notice these are feeding into these points. Tropomyosin is related to a contractile protein, band 4.1. So you can see these little points here where there is, they call that a junction complex. So this whole structure here is called a junction complex. And that's in binding and sending the spectrum dimers throughout, maintaining the biconcave nature of the structure here. So there's a lot of protein-protein interactions that we could test in here. Here's the anchoring molecule. Here's actin at these structure, at these junctions. Here's the spectrum here, alpha and beta. It's kind of hard to see them twisted. And these junction points in between the junctions, here's a junction, here's a junction. There are anchorings that anchor the spectrum here, okay, and keep it firmly in place. So there's a combination, a complexity of structure here. So are proteins mobile? Some of the earliest work where they would take a fluorochrome that's green in mouse cells, and human cells, proteins were labeled in red, and actually fuse them together using a virus fusion protein, hemagglutinin from flu virus. And what they saw is in the beginning, about half the cell was green, the other half red, and over time, notice the mixing. The cells became red and green after 40 minutes. So we know for sure that proteins do move throughout. We talked about lipid rafts and how those retain certain proteins, but just in general, there's a lot of fluidity to proteins in the cell surface. Hydropathy analysis and how is it used? We hinted this with some of our structure, and there are bioinformatic software that allows you to plot hydrophobicity or philicity, and if you're hydrophobic amino acids, they will end up being plotted above 0. Hydrophilic, water-loving amino acids will be plotted below. So we got hydrophilic and hydrophobic domains of this one protein. So this protein spans the lipid bilayer 4 times, and then there are especially key areas that are inside, and it's probably where there's other interactions to occur, and they show you the structure here of the interactions. Okay. So any protein can be tested and looked at that, and you can get that structure. So how do we determine whether protein is exposed to the surface or not? The first experiment was using an enzyme known as lactoperoxidase. It's where proteins containing tyrosine that are on the cell surface would be labeled with radioactive iodine 125. So this enzyme, in the presence of the radioactive iodine, will label proteins. So notice we have a cell, and we have proteins A, B, and C. Notice A is peripheral on the outside, C is peripheral on the inside, and B is trans. They do the lactoperoxidase with the I-125, and A becomes labeled, and this part of B becomes labeled. C is not labeled. So if you dissolve these now and looked at them, and then exposed to X-ray, A and B would be lit up. C would be found in Kamasi staining, but would not be found in radioactive label and detection on X-ray film or phosphorimager. So right away we would know A and B had parts exposed to the outside, and C was completely protected. So it's kind of a protection assay. If you swell the cells up, like red blood cells, and then you add lactoperoxidase, you put the lactoperoxidase inside, you then reseal and add I-125. B and C are labeled, but A is not. So now, if you carry the experiment further, and this was the original paper where they did that in 1973. So A is on the surface and not inside. B is on both outside and inside, and C is on inside. That's what that data is telling you. So now we can compare, did a protein, is it a transmembrane, is it peripheral? That it shows that. And so this was the original technique where they were looking at cells, and that's where they got the lactoperoxidase. So not only can be proteins, through the membrane or associated with either leaflet, but they have carbohydrates that can be attached at asparagine residues. And there are also O-linked sugars. It's serine threonines. So proteins can have N-linked asparagine, O-linked, which is serine, and threonine. And it just shows the different structure of the N-linked and the O-linked here, these two. And we talked about proteins a little bit. This is the complex form. Okay, it's made it all the way through to the transmembrane. It's processed completely. Here's asparagine. Here it is, and here's the simple form in the ER. Okay, lots of mannose, glucose. And so we've talked about how there's a fatty acid carrier called dicol that the sugar tree is built on, and then an enzyme will detect the asparagine it's exposed, and using the energy built in with the phosphate, attach the sugar to the protein that's being made in the ER. This is the rough ER. All sorts of different sugars. In biochemistry, you'll have to memorize all these structures and whatnot. Galactose, mannose, NAG, and sialic acid. Here's some more. Not you have to memorize them. Here you will for biochem. So here's one of these serpentine receptors, rhodopsin. It's involved in light pigmentation here. Shows a hydrophobicity profile. Showed the hydrophobic and the hydrophilic regions. And then here's the actual structure. And so coming into the ER, there are start and stop areas that allow this to be kind of threaded through here. And so there are domains here that have information for translocation machinery. Okay. So how would we figure out, is this the stop? Well, it's going to be hydrophobic. If you got rid of it, then you would not have this structure. Okay, it'd be a different structure. This is just showing some more structure of the, here's the junction, the spectrin, the ankrins are here. Okay, and just go into more detail how they figured out all this was a complex. You could see some of it, you could purify some of it, a lot of interactions here. So when you do an STS page of red blood cells after treatments, and identifies soluble, peripheral, and integral. So here's our red blood cell, and we centrifuge. And so we have total protein here. And if you run that here on this lane, you get all the main proteins shown here. If you rupture and hypotonic, any proteins that are soluble, okay, will be gone. so you're losing the red protein. And under pH 11 carbonate conditions, you will start stripping off, okay, peripheral proteins. So you can strip them off. You can rupture, soluble, strip off, peripheral, and then of course Triton will get everything's gone except the membranes, and it will take the membrane proteins out. Here they is. So if you do a ruptured ghost and look at the proteins left, these are the membrane proteins, either trans or peripheral. If you do pH 7 carbonate, you find only the transmembranes here. And when you add detergent, nothing spins out because you've released all the proteins. So now under these conditions, you can run these. Here's a ghost. Notice red is gone. And then the pH 11 carbonate, they call it pH resistant right here. So in these three lanes, we know the total proteins. We know this is soluble, and we know these two are membrane associated, membrane transmembrane, and therefore we know these proteins are peripheral on the inside. So this is how they break apart the structure there. And then if you do protease protection, here's your red blood cell, and you chew up with protease. Notice it chewed back all the proteins, and then you add detergent, nothing left. So here's the red blood cell. If you add protease, notice you get this structure, and what's happened is these two right here, right there, the purple and the blue have decreased in size. The only way that could have happened is they would have been partially chewed. And with detergent, you see nothing at this point. So protease protection implies all these are protected. Okay, protected. Okay, protease protected. Okay, these are sensitive. Okay, they've been chewed. Right here, protease. Here's 1995, this paper. The MALK protein of the ATP binding cassette transporter for the sugar in E. coli is accessible to protease on the periplasmic side. So now they figured out this transporter protein, and protease can get to it. So they do proteolysis of right side membranes, and they digested with protease proteins here, and they were digested for probably 0, 3, 5, and 7 hours here. So here's a time here. Okay, notice almost all proteins chewed away here. Here's a control. It's protected the whole time. Okay, control. Okay, control. And then here's the experimental. So you can plot these out and figure out amount of protein. Here's the amount, MALK getting chewed. These other proteins are stable, protease protected. So the first big study on plasma membrane proteins was red blood cells, because they're easy. Red blood cells, when they're fully mature, have no organelles, no nucleus, nothing. They're pretty much stripped down to do that. So the plasma membrane is only membrane the red blood cell, so you don't have to worry about any contaminating membranes. Here they are. Okay, here they are. Here's our ghost concept, and now we have right side and inside vesicles. We can isolate them. And cytosol inside is hemoglobin. So hemoglobin's sitting there, and when you open them up, the hemoglobin's released And so there's no hemoglobin in these right or inside vesicles here, right there. No hemoglobin. So you label some type of fluorescent or radioactive. We talked about I-125 and lactoperoxidase. Whatever you're labeling has to be water soluble so it won't go back through the cell. So you can label the right side vesicles and then run an STS page and see what proteins are on the outside. Okay, and this just shows the separation after the sealing of the right side vesicles, right side, and it's stained with Kamasi blue, and it's the drawings here. Is exposed to the right side, so they found glycoporin is sensitive on the outside there, on the outer surface. Here's inside vesicles. Glycoporin is labeled. It's exposed in the inner surface as well. Thus, a type of membranes can be discerned. Here's glycophorin. So some of it's on the inside, some of it's on the outside. So it could be labeled on either side. It's a transmembrane. That's exposed to the outside, exposed to the inside. There were 6 major proteins, many more, but those are the 6. Spectrin, anchored, here's spectrin, long flexible rod, dimer, we talked about that. Here it is, the structure, heterodimer, 2 anti-parallel peptides, alpha, beta. Self-associate, they interact head to tail, and you can find what it looked like. Spectrin, it's 4 of these come together and joined by actin filaments here. Here's band 4.1, and so tetramer and actin. Tetramer's length, that's the junction complex, and the protein mesh underneath. The reason you need the mesh and the shape, the stress of these going into the smallest capillaries, they had to squeeze themselves through. Okay, so we talk a little bit about labeling, sidedness, protease protection, figuring out what's on the surface, what's peripheral, what's trans, what's cytoplasmic, all that here. And this is kind of that same concept where you fuse a human in a mouse cell. They have different membrane proteins that you label, have different antibodies, and you notice first labeling, there's the red side and green side, and then over time, 40 minutes, they become both red and green. So there's another technique called FRAP, which you radio labeled all the surface, and you actually zap a region of the lipids, and if you hit the fluorochrome with enough laser beam energy, you will bleach out and destroy the fluorochrome's fluorescent, and so you end up getting a non-fluorescent region. If you allow recovery, what they will see over time because of fluidity is membranes that are labeled in outside the bleached area will move in. And this is just shown the graph showing the recovery time. So each cell, therefore, can be measured on their recovery time. So now we can measure how good your cells are to moving membranes around, membrane proteins. So now we have another personalized parameter that when they isolate all that, they'll be able to figure out just how efficient your cells is compared to somebody else. Is there any aspects of disease that are just due to the simple nature that your cells are a little slower in moving material around the plasma membrane? Yeah, it never ends. And so here is a laser, you bleach it, and then you start measuring, and you do continuous bleaching. And over time, this area you're measuring drops, and it shows that bleaching will drop that area. Okay, so it's not like all these proteins land here, everybody's getting bleached. Okay, I think we've talked about this, constitutive regulative. And then the biosynthetic pathway is here to the endosomal lysosomes. Golgi, the stacks, early endosome, we've seen this as well. So it turns out that there's a lot of fusion events of material coming in and out of the cell, and you can actually measure all these little red Roman numerals here are aspects we can actually measure your cell's efficiency for moving material in and out of the cell. Very interesting here. Here's epithelial cell. So when they started looking at membranes and vesicles moving, okay, remember the Balch experiment with the Golgi fractions? Well, in some way, he was getting into membrane trafficking. It's a lot of hard work to isolate the vesicles from different donor and acceptor compartments. So a lot of researchers were working in yeast and using genetics of yeast. Yeast are haploid, so they have one copy of each gene. Okay, one copy, haploid. Okay, can't spell. And they were looking at yeast that were defective in moving vesicles around, and they called those SEC mutants, S-E-C, or secretory mutants. And so Sheckman and his work were done by looking at vesicle transport genetically in the cell, and Rothman and Balch were doing inside the cell-free system. So you had two groups working in an in vitro and in vivo systems here. And again, set up the system, Balch and them looking at between the compartments. And Balch also went on to do ER to Golgi transfer. And we've talked about, this is a slide we've talked about following that up. Okay, so, and what they found was if you added ATP and cytosol, okay, you would get vesicles fusion, and the protein got the right sugar incorporated, it's tritiated. If you just added ATP by itself and no cytosol, you got no movement of vesicles. So energy is not the only thing required. It's something in cytosol required. Then they took cytosol and treated it with various inhibitors. One inhibitor was NEM inhibitor. This chemical modifies cysteine amino acids and inactivates any protein that has an active cysteine. NEM. And so what they found was when they fractionated the cytosol, plus or minus EM, they found there was a protein that they named NSF, N-ethylmalamide sensitive factor. So this protein was being acted on by that inhibitor. This protein is a monomer. It's an ATPase. It forms oligomers in a test tube, and NSF is required for multiple points in the secretory pathway, not just through the Golgi. So here was a common piece of protein machinery that was involved in making vesicles move between compartments, and it could be inhibited by NEM, which will shut down cysteines. So this protein, NSF, must have cysteine residues that this chemical blocks and binds and blocks and shuts down its function. So a lot of research is done with chemical inhibitors.